This presentation is on multiple sclerosis and its mechanisms, management, and ethical considerations. So, what is multiple sclerosis? Multiple sclerosis is a demyelinating disease of the central nervous system, which includes the brain and then the spinal cord. The myelin sheath is a protective sheet that surrounds the axons of the neurons, allowing them to quickly send electrical impulses. This myelin sheath is produced by oligodendrocytes, which are a group of cells that support neurons. In multiple sclerosis, demyelination happens when the immune system inappropriately attacks and destroys the myelin, which makes communication between neurons break down and ultimately leading to all sorts of sensory, motor, and cognitive problems. Now, the primary cause is unknown, but we'll look at the pathology later on. Now, why is it important to learn and understand this disease? To answer that, we must look into the prevalence and the impact it has on the society. The disease varies between age, sex, nations, and even genetics. It is important because the statistics suggest a high prevalence of about 2.5 million people are affected by multiple sclerosis, while about 400,000 of them are in the US. Although it is more common within the European populations, the effects are still significant. In terms of sex, females are most likely to be affected by multiple sclerosis than males within the same age group, and it is higher in women in their 40s. There are also genetic factors that contribute to the disease, such as family history would increase the risk, and also the HLA DRB1 allele. There are other environmental factors that would contribute as well, such as low vitamin D, smoking, Epstein-Barr virus, in short, EBV virus or infection. And these data are supported by studies such as a study conducted in England, which I'll show in the next slide. So the data from the last slide are supported by the findings summarized in this slide. First, in this graph, we can observe that multiple sclerosis rates are again higher in female populations, which is shown in the teal bars, compared to males, which are in the beige bars. And seeing that in both populations, the numbers are significantly high. Next, we can also see the focus on the beige bars over here, which constitutes of ex-smokers, and then the darker ones are non-smoker, and teal is current smoker. And looking at it again, we see female ex-smokers have higher percentage, higher rates of getting multiple sclerosis than the males. And if we focus on the non-smokers, such as female non-smokers, still have higher prevalence than the male non-smokers. And lastly, a comparison between the UK and its neighboring countries. As we can see, UK has the highest prevalence of getting affected by multiple sclerosis than the neighboring countries. Speaking of pathophysiology, the brain, including the neurons in the brain, is protected by things in the blood called by the blood-brain barrier, in short, the BBB, which is this is the blood-brain barrier. And this blood-brain barrier lets only certain molecules and cells through from the blood into the brain. And for immune cells, like T and B cells, that means having the right ligand or receptor is required for it to get through the blood-brain barrier. Once a T cell makes its way in, it can get activated by myelin. And once it gets activated, it changes the blood-brain barrier cells to express more receptors and then allows more immune cells to bind and get through easily. As we can see over here, the T cell is squeezing through the blood-brain barrier, and it interacts with macrophages. It also interacts with macroglia. After that, um, the multiple sclerosis is type 4 hypersensitivity reaction or cell-mediated hypersensitivity. This means that the myelin-specific T cells release cytokines such as um, the IL-1, IL-6, as well as TNF-alpha cytokines and interferon gamma. And together they all dilate the blood vessels, which allows more immune cells to get in. And this will directly cause damage to the oligodendrocytes, which are these oligodendrocyte cells, and so on. The cytokines will also attract B cells and macrophages as they're part of the inflammatory reaction. Um, and then, as a result, those B cells begin to make antibodies that mark the myelin sheath proteins. And then the macrophages use those antibody markers to engulf and destroy the oligodendrocytes. As we can see here, the macrophages are attacking on the oligodendrocytes. And without oligodendrocytes, as we know, there is no myelin to cover the neurons, and this means this leaves behind the the axons to be exposed, and this would sometimes lead to will lead to scar tissue, also called as plaques or sclera, and then an autoimmune attack on the oligodendrocytes will then regulate the T cells to come and inhibit or calm down the immune cells, leading to reduction in inflammation. And early on in multiple sclerosis, the oligodendrocytes will sort of heal and extend out new myelin to cover the neurons, which process is called remyelination. But unfortunately, though, over time, as the oligodendrocytes die off, the remyelination would stop, and then the damage becomes irreversible, of course. And then this loss of axons kind of make the uh, neurons to be more prone to the sodium calcium influx, as the exposed axonal membranes would kind of be exposed to the sodium calcium influx, and then it would cause mitochondrial dysfunction. And then over here in this schematic, it's focusing on lymphocyte recruitment, neuroinflammation, and axonal damage. Here we can see first is the peripheral immune activation, which is by the CXCL13, which is a chemokine, and it is secreted by beta cells and dendritic cells in the lymph nodes. And what it does is basically recruits B cells and T cells via the CXCR5 into the central nervous system by creating a chemotactic gradient. And this kind of links to multiple sclerosis by elevating the CXCL13 in CFS or the cerebral spinal fluid, and it correlates to multiple sclerosis relapses and lesion activity. There is also the T cell receptor, in short TCR, over here, and then the Th17 cells, which are kind of like a pro-inflammatory molecules and are activated by the periphery myelin antigens, or in short, there is also myelin-based proteins activate these molecules. And then these cells kind of produce this molecule again, which is a cytokine again, I mentioned in the previous slide, IL-17, and it kind of breaks the immune tolerance. And these molecules, such as the Th17 and B cells, migrate across the blood-brain barrier, and then they secrete matrix metalloproteinases, or in short, MMPs, and degrade the tight junctions. And then this molecule IL-17 also further disrupts the blood-brain barrier by inducing an endothelial inflammation. So this all of these collectively kind of causes inflammation in the CNS as well as demyelination, and these IL-17 are also kind of drives reactive astrogliosis, or basically where um, astrocyte and feed hypertrophy as well as microglial activation, and it also amplifies neuroinflammation. um, and then there is also, as we can see, there is in plasmocytes are also involved. And then there is also osteopontin, or in short, OPN, as we can see here. These are also a pro-inflammatory cytokine, and they enhance the T cell survival and inhibit remyelination by blocking oligodendrocyte precursor cell, which is OPC differentiation. So by this means, through this, that means there is no remyelination in the axons and they're very exposed. And then over here in this image kind of summarizes the immune-mediated attack on myelin. As we spoke in the previous slides, basically there is the involvement of the Th1 and Th17 cell, as well as the humoral B cell or antibodies are also involved. And this means the CD4 Th1 cells are activated by myelin antigens over here, and then they secrete the IFNY, and then this activates the macrophages, which attack myelin via phagocytosis, and then it releases TNF alpha cell or molecule, which directly damages the oligodendrocytes over here. And then this results in focal inflammatory lesions with myelin destruction. And then there is the other pathway, which is the humoral immunity, where we see that the beta cells kind of are involved in it, that produce myelin specific antibodies such as anti-MOG or anti-MBP. And then from the um, oligoclonal bands, they um, they produce antibodies. These antibodies bind to the myelin and trigger complement activation and attack the membrane complex. Yeah, and then this causes again all these both of these pathways collectively attack the myelin sheath as well as the oligodendrocyte and causes demyelination. And as terms of symptoms and diagnosis, the symptoms vary depending on the location of plaques, and this may worsen over weeks and then kind of linger around for months without diagnosis. And these are some of the diagnosis that can be used. First of all, there should be medical history taken, such as looking for diabetes, thyroid disease, food habits, and so on. There is also geographic locations take part into this, and substance abuse. There are also neurological examination that could use imaging techniques such as the MRI, which is very useful in this, which identify any scar tissue and um, formation of the scar tissue, and then the damage in the CNS, such as looking for lesions, as we can see in this picture. And then there is also lumbar punctures, in short LP. There is also evoked potential test, which kind of measures the demyelination in the optic nerve, and it also checks for response to visual stimuli, and of course, blood sample analysis.